Video we'll be looking at the non-lactose fermenter in the family Enterobacteriaceae, primarily Salmonella and Shigella. Main difference between Salmonella and Shigella species is the prevalence of H2S producing species in the genus Salmonella. So Salmonella was first discovered by Theobald Smith in 1885, and he named the bacteria after his partner Daniel Salmon, a veterinary pathologist. In the past, Salmonella taxonomy was complicated because it was based on serology via the somatic and the flagellal antigens, and the taxonomy ended up with more than 2,000 species, 2,000 serovars or in Salmonella. So it was a very difficult task to identify Salmonella. Now we only recognize two species in Salmonella, which is Salmonella enterica and Salmonella bongori. And at present there are more than 4,400 serovars in this genus. So Salmonella enterica, subspecies enterica, serovar typhi, is the actual name for the conventional Salmonella typhi. And if you look at the typing here, the typesetting here, only the genus is italicized, whereas typhi is not. So this is only acceptable for Salmonella. So in Salmonella enterica is further divided into 6 subspecies: Enterica, Salame, Arizone, Diarizone, Putinae, and Indica. So among these 6 subspecies, Enterica is found in the intestinal tracts of humans and many warm-blooded animals, whereas the rest are from cold-blooded animals and the environment. Most of the bacteria that is found on human hosts that causes diseases are from the subspecies Enterica. So this shows the utilization of the serovar O antigens and the H antigens to type selected Salmonella species serovars. So there is also the VI antigen for Salmonella typhi, which is actually a capsular antigen. We can divide them into typhoid salmonellosis and enteric salmonellosis depending on the diseases that they cause. So for example, the serovars that cause typhoid salmonellosis are typhi, paratyphi A, B, and C, whereas for enteric salmonellosis they are mainly enterotitis and typhimurium. Infection spectrum: so typhoid salmonellosis only infects humans, enteric salmonellosis both animals and humans, and the source of infection only from humans here and livestock and sometimes humans. The mode of infection is similar, both oral through contaminated food and water. Infective dose is very different. Very small infective dose for typhoid salmonellosis and a very large infective dose for enteric salmonellosis. So that would explain why the incubation time is a lot longer for typhoid salmonellosis, because the bacteria needs to multiply and grow further before the infection starts. whereas for enteric salmonellosis, the incubation time is shorter. Because typhoid salmonellosis also invades the lymphatic system and then the bloodstream, it causes generalized infection with sepsis. For enteric salmonellosis, there is no invasion into the lymphatic system or bloodstream. So what you get is acute diarrhea with vomiting, with fever. And this in enteric salmonellosis is usually self-limiting, which means even without therapy, it will go away. So this is the diagnosis method, the types of therapy recommended, and also how to prevent. So for typhoid salmonellosis, as mentioned by Salmonella typhi, paratyphi A, B, C, only human carriers. What is important here is invasion by endocytosis via the intestinal tract, where they continue to enter lymphatic tissue and then enter the bloodstream. Okay. So this mode of infection is important and differentiates Salmonella from Shigella. So for typhoid salmonellosis, you require antibiotic therapy, but the problem is a small percentage of the patients will remain as chronic stool carriers. That is why for food handlers in Malaysia, there is a regulation where it is compulsory for all food handlers to be tested for Salmonella to see whether they are carriers for Salmonella. Mary Merlone is the famous typhoid Mary, an asymptomatic carrier that infected up to 53 people in New York, USA. She worked as a cook, and it seems that Salmonella typhi was in her gallbladder. For enteric salmonellosis, the infection source is usually livestock, mainly contaminated whole eggs, meat products. Pet reptiles are a source because most of the pet reptiles, up to 90%, carries Salmonella, but the infective dose is very high. So if you are careful, you shouldn't be infected by it. In the past, raw eggs was part of the menu in Japan cafeterias. However, because of salmonellosis, they removed the menu from all campus cafeteria. So the oral infection through food and water before attachment to enterocytes of ileum and colon, then invasion into the ileum. There is persistence and then enterotoxin production. So enteric salmonellosis will abate after several days. And for diagnosis, usually the sample is stool, where you have to culture in either McConkey agar, both McConkey agar and Salmonella Shigella agar. If for carriers, as the number of Salmonella can be low, an enrichment step with Senilite broth or Rappaport vacillidis is recommended. So the difference here, Salmonella is motile, whereas Shigella is not. So this is very important to differentiate them. Another is most Salmonella produces H2S, Shigella does not. Okay. So for dried food products, to isolate and identify Salmonella, there's a pre-enrichment step, lactose broth at 35 degrees Celsius for 18 to 24 hours. There is also a selective enrichment step, usually tetrathionate broth plus selenite cysteine broth, or tetrathionate broth plus Rappaport bacillarius broth. Although Rappaport bacillarius is not really suitable for Salmonella typhi, it is safer than selenite cysteine, and therefore this pair here is actually recommended over this. So, however, if you are specifically looking for Salmonella typhi, then you can use this pairing. So you're supposed to use both the enrichment broth. However, please note the incubation temperature for tetrathionate broth is 35 degrees Celsius, whereas for RV is 42 degrees Celsius. After incubation, you can then carry out streaking onto selective plates. You have to use at least 2 types. You can choose from brilliant green, bismuth sulfite, Salmonella Shigella, McConkey, desoxycholate citrate, HEA, or even XLD media. So this shows the colony morphology of non-lactose fermenter versus lactose fermenter McConkey agar, non-lactose fermenter versus lactose fermenter on HEA or Hectone Enteric agar. Here you can see that this Salmonella species here produces H2S. That's why the colony is black. So once you have a presumptive Salmonella colony, you have to purify them and then carry out biochemical identification tests, either by separately, or you can use a multi-test kit. Then final confirmation is via serological tests. Okay, so these are some of the for confirmation results for Salmonella identification. The genus Shigella, this genus was named after its discoverer, Kiyoshi Shiga, about 100 years ago. So there are 4 species here: Shigella flexneri, Poidy, Sonnei, and Dysenteriae. All causes bacillary dysentery or shigellosis. Okay. This is primarily a disease of young children through contaminated foods, water, or unhygienic, dirty hands entering your mouth. Okay. So the source in each case is usually unwashed hands, okay, where the primates are the only reservoir. The infective dose is very small, from 10 to 200 cells, and because this bacteria can withstand stomach acid, it grows in the small intestines but attacks the large intestines. It invades the epithelial lining layer but does not penetrate. So this is different. This characteristic is different from Salmonella. So it will grow in the epithelial cell and release the Shiga toxin that results in the damaged epithelial layer. That is why in your stool there is a lot of mucus and blood. So you can see that the Shigella is able to multiply in the cell, and then it can also invade neighboring cells, but it does not go further into the lymphatic system or the bloodstream. When the toxins are released, the cells are damaged, abscess is formed and you can see blood and pus in your stool. So Shigella-like toxins or verocytotoxins, they inhibit protein synthesis, and you can see bloody species after incubation period of 2 to 5 days, a lot of intestinal pain, a lot of bowel movements in the day, sometimes you have fever, but usually resolved after 5 to 7 days. However, with the loss of electrolytes and water because of the high number of bowel movements per day, usually therapy is recommended, and the diagnosis is by plating stool samples onto McConkey agar, Pectone enteric agar, or XLD agar, where you look for non-lactose fermenter. So here you can see between these two non-lactose fermenter on HEA, this doesn't produce, Shigella doesn't produce hydrogen sulfide. So there is no black precipitates on its colonies. Look at other Enterobacteriaceae that are important, especially those causing opportunistic infections or hospital infections. So here we have Citrobacter, Klebsiella. So these are lactose fermenters, so they will form pinkish to reddish colonies on McConkey agar. For Klebsiella pneumonia, they have a very unique characteristic where they form very mucoid colonies on artificial media. This is because they have this polysaccharide capsule that gives them this appearance. So they are important in nosocomial infections or hospital infections. Enterobacter and Serratia are also lactose positive Enterobacteriaceae that causes hospital infections. Next we have Proteus, Morganella and Providentia. So these are lactose negative Enterobacteriaceae. So for Proteus, they have a characteristic of swarming on nutrient agar and other artificial suitable artificial agar. So this is how swarming looks like, where they swarm to form concentric circles moving out from the point of inoculation. CA, a different family under the order Enterobacteriaceae. So the famous bacteria genus under the family Yersiniaceae is the Yersinia pestis, Yersinia enterocolytica. So Yersinia pestis causes the bubonic plague, whereas the Yersinia enterocolytica causes enteritis Yersinia. So Yersinia is also lactose negative, so they will form pale colonies on McConkey agar. Thus, they are urease negative, so this helps separate them from Proteus, which gives the urease positive reaction. They are also indole negative. So Yersinia pestis is the causative pathogen for the plague. So in the Middle Ages, it is known as the responsible for the pandemic, the Black Death, that killed from 30 to 60% of Europe's population. So this was primarily around this period. The town population was increasing without any planning for sewage disposal, proper sewage disposal, or rubbish disposal. So this inevitably caused the rat population to increase, and as you can see later, how an increase in rat population correlated with the increase in infection from Yersinia pestis. So infection occurs when there is a direct contact with a diseased rodent, a rat that has a Yersinia pestis. So the Yersinia pestis will enter through the skin and reach the lymph node, where they grow, forming bulbos. So this is a bulbo. At the second stage, the Yersinia pestis enter the bloodstream, then infection spreads to other organs. For diagnosis, isolation and identification from suitable samples, you will see short gram-negative rods showing bipolar staining. So you can see the stain is heavier at the poles of the cell. So this shows the life cycles that are known for Yersinia pestis. So you have this zoonotic reservoir where Yersinia pestis survive on infected fleas on wild rodents. However, sometimes the infected fleas migrate to rats, and these urban rats continue to propagate them. And when dense living quarters, poor hygiene sometimes will facilitate the transmission of infected fleas biting humans, and when they bite, they will transmit the Yersinia pestis onto humans. So initially it will cause bulbos. At this stage, first stage of infection, you'll have the bubonic plague. However, once it spreads to other organs, especially lungs, you'll have bacteremic seeding of lungs where cough, respiratory droplets all contain bacteria. Then this becomes the more contagious stage of pneumonic plague, which is why contact persons must be quarantined for 6 days, because it is easy to spread when it reaches its pneumonic stage. Therapy requires antibiotics and 100% mortality rate if untreated. So other than bubonic Yersinia, you also have enteritis Yersinia. This is caused by Yersinia enterocolitica and Yersinia pseudotuberculosis. They also have reservoir in wild animals and livestock, and infects human who takes up the pathogens in contaminated water or food. These organisms penetrate the mucosa of the lower intestinal tract and causes abdominal pain and enteritis. So Yersinia enterocolytica is responsible for 1% of acute enteritis cases, especially in Europe. Okay. Yersinia pseudotuberculosis is less significant and usually no chemotherapy is required.